So we're going to start a new unit today that everything in this unit is directly relevant to how membranes behave. So what they let cross through them, what they don't let cross through them, how are proteins structured within a membrane, and what are membrane properties. So after talking about protein structure, membrane structure and function is probably the second most important topic that goes through this whole class. I need to actually open this slide so I can click through. Skip over that. Okay. We'll come to that later. So I know that in previous classes you've learned that membranes are semi-permeable boundaries between two solutions. And that is true. So if we think about the membrane that you've talked about the most, the plasma membrane around the outside of the cell, the properties of that membrane allow the cell to do homeostasis. So if you want to keep the inside separate from the outside and different from the outside, letting different things through the membrane or not will let you maintain that environment. And that concept is also true for membranes around organelles. So every time you have a new membrane-bound compartment, that compartment has the potential of maintaining different conditions and different sets of molecules than what's on the other side of that membrane. So in this class, we're going to more often look at three compartments at once and how they differ and how they behave, so to speak. So to do what the cell needs, the membrane has three big jobs to accomplish. And this would also be true for internal membranes, but for simplicity, they're just showing the plasma membrane. Import and export is the one I just talked about. So what's allowed in, what's sent out. This would be more like border control type of capacity. This, having a border that lets some things through and not others, sort of begs the question, well, how do you get information in and out? How do you have a barrier that also can transmit information through it? And so this factor of membranes largely, not totally, but largely relies on proteins that cross or associate with the membrane. Sort of like having a person stationed at a border that's maybe on both sides or maybe within the barrier, getting information, sending information. A lot of our signal transduction pathways that we'll look at later, they start with something that happens outside the cell. Something, get the information to the inside of the cell. And the third function is related to how can the cell move? How can the cell move or grow and still maintain a boundary? This one I don't talk about as much in this class, and it's not in this unit, and it comes later. So it must be able to send and receive signals, control passage of molecules, and also let the cell expand and grow or move. It has to accomplish all of those things. And I know that you've learned before that the plasma membrane is a fluid mosaic. It's totally true. Fluid because it moves. It moves quite a lot. and mosaic because it has different proteins and other molecules associated with it. And most books don't show enough mosaic in the fluid mosaic. So membrane, if you take a piece of membrane from a random location in a cell, it can be 50% protein and it's quite crowded with protein. If you remember the video that we watched the first day, how the membrane lipids were constantly moving, like more than an ocean. And then there were proteins that were moving and quite a lot of motion and jostling and squeezing past each other in this fluid mosaic. I'll show that video again after the break today. I know you have also looked at membrane lipid structure before, and this is going to be a little bit of a review. And recall that these, just point to it, membrane lipids have an amphipathic structure. What does that mean?
That means part of it's hydrophilic and part of it's hydrophobic.
Yes, part of it is hydrophilic on one end, hydrophobic on the other end, amphipathic. And that lets membranes have an exterior and an interior. So for these phospholipids, the polar groups or the head groups, these are oriented next to water, and the tails are oriented next to each other in two leaflets. So let me come back to the slide in a minute, but I want to go onto this. So recall from earlier, we talked about the reason that anything is hydrophobic is that it forces water to do caging. You remember that? So we could have a nonpolar molecule like this, and water has to break its hydrogen bonding network with itself and do caging around it. So the hydrophobic parts of phospholipids will naturally associate together because their association, the association of the hydrophobic tails increases entropy overall. So this is one of the great ordered looking structures in nature is a lipid bilayer. And what you're looking at here, these are water in a ball and stick form. This is water. And this is one leaflet of a plasma membrane, the other leaflet. And yeah, this is ordered. They're lined up with tail to tail. But overall, that is, it increases entropy overall because water does less caging. That fact and the fact that membrane lipids are amphipathic is really, really good for cells for two different reasons. The first reason that this is great for cells is that if you put lipids in water, phospholipids in water, you do not have to do anything special to get them to form a lipid bilayer. And many of them, depending on their concentration, will form various sizes of vesicles. So that fact about increasing entropy overall, that means, therefore, that vesicles can form spontaneously. It's not hard for a cell to make a vesicle form. It's an entropically favorable process to have a sphere, so imagine it in 3D, a sphere of membrane lipids with two compartments on either side. This also has implications for how life could ever form. Like, how would a cell ever form? If phospholipids could form, membranes can self-associate in an aqueous environment. And it's also useful for cells today not to have to spend energy to make vesicles. The second reason that this amphipathic nature and self-association is great for cells is if the plasma membrane rips, if it sustains damage, and if the damage is not totally catastrophic, free edges will self-seal, which is cool. Like if you took a tiny probe or something and you were to stir around, let's say, the endoplasmic reticulum inside a cell and break it up, it would self-seal and still function to a certain point. If you put a tiny blender in there, no, but it would self-seal to a certain point. So this property also allows cells to fix their membranes without adding extra energy. It doesn't say that if this happened and it self-sealed, the cell would just suffer no consequences from that, but membranes self-seal and are in some ways self-healing if the damage is not too great. So the next thing I want to talk about is how membrane lipids move and how membranes generally move. Lateral movement is frequent. Lateral movement of phospholipids is frequent. So this is like if you're in a crowd at Rhythm and Roots, you can squeeze by, wedge past people, make your way from here to here with some jostling, and individual phospholipids will move in that way laterally. They also can flex their tails. Thermal motion will just cause that to happen. And they also can individually spin around. It's kind of like if you're standing in a crowd and I'm tired of standing here, I can move over. I could like spread my leg out and get more space. I could lean this way, lean this way. I would not personally spin around very often, but I could if I wanted to. So those kind of movements give you the fluid part of fluid mosaic. Quite a lot of movement. I want to go back to something that has to do with tail kinks as well. So yes, we have hydrophilic heads associated next to water and tails lined up tail to tail in a hydrophobic region. But notice how they have different levels of kinkedness, so to speak. Have you ever talked about this? Saturated, unsaturated? Okay, now I'm not going to go through it in detail, but just recall that if you've got a kink, that leaves more space between you and the neighbor, and there's more room to move. You recall that? Because of the double bonds. Let me go back and just show it. Every place where the hydrocarbon chain is double bonded, there's a kink. And we call that unsaturated. And unsaturated hydrocarbon chains, the phospholipids that have those give greater fluidity to the membrane. So if I use the crowd example, I would hope that my leg is not sticking out like that. But if you've got a big purse or a bag that gives space between you and somebody else, you've got more room to move. Another thing I wanted to draw on here is the concept of leaflets. Make sure that's on 2 slides away. Come to that in a second. So think about, all right, we have space between different phospholipids, and if they have greater kinks, they have greater fluidity. So extra double bonds in the hydrocarbon chains increase fluidity. Cholesterol also affects fluidity of the membrane. And this is the structure of cholesterol. It's derived from steroid hormones are derived from cholesterol. It has this particular 4-ring structure and a hydrocarbon tail. And if you imagine, I have a phospholipid right here and it has a tail that kinks like that. This is the perfect shape to fill kink space, for example, like here. Kink space is not the term for this, but if we think of that as the space provided by a kink in the tail, cholesterol fills it and wedges in there. So cholesterol has the opposite effect of unsaturated bonds. So whereas unsaturated bonds increase fluidity, cholesterol decreases fluidity in a membrane. And so those two factors can help a cell change its membrane fluidity if it needs to. And organisms that live in really cold places have really, I'm talking about microbes, they have really a lot of kinked tails and less cholesterol because they need to keep their membranes moving at super low temperatures. Now we can talk about leaflets. Way up for this. Any membrane that we talk about that's a living membrane has two parts. So yeah, it's a lipid bilayer, but each half of it specifically is called a leaflet. So the leaflet is the half that contains those phospholipids and those cholesterol molecules and whatever is stuck on the phospholipids. That's the leaflet. So if we take these labels seriously, that this is outside the cell and this is interior to the cell, we would say that this is the outer leaflet and this would be inner leaflet. Or you could name them cytoplasmic leaflet, extracellular leaflet. Okay. That's the layer. When we talk about the face of a membrane, that's like being outside of it and looking across the 2D surface. So if I were tiny and I could look toward this leaflet, I would be seeing the cytosolic or the cytoplasmic face. It's sort of like, are we talking about cross-section or looking straight on? So this would be the cytoplasmic face, this would be extracellular face. And any given membrane does not, it's two-faced. It's two-faced. It's got different sugar groups hanging off, especially the extracellular face for mammalian cells has particular sugars that hang off of it, and our immune system see those and say, that's me, that's self, I see my marking sugars. And the cytoplasmic face has different character as well. So it could be different chemical components of the head group and also sugar attachments onto those, but they have totally different, like you could recognize them if you were a tiny molecule. Oh, that's interior face, that's extracellular face. And this would also be true for organellar membranes. Like the inside of the mitochondrial inner membrane is different than the intramembrane space of the mitochondrial inner membrane. They have different characteristics. So, like any molecule, membrane lipids don't last forever. They have a lifespan. And if we think about how lipids get to and from membranes, they travel in vesicles. So that's worth writing on here. Membrane lipids travel in vesicles. So the lipids themselves travel between membranes or compartments in vesicles, or as vesicles. They are making up the vesicles. And the place where cells make membrane lipids is in the ER. So if you think about ER as this large blobby network of membrane, the inside of it is the lumen. That's an important word to know. The inside of the ER is the lumen. And outside of that is the cytosol. So the lumen has enzymes that make phospholipids. One more. The lumen has enzymes that make phospholipids, and they are made on the lumen side, and they go and join the cytosolic leaflet. So it's a little bit counterintuitive. They're made on the inside, so it would be here. They join the cytosolic leaflet. Part of that is to provide natural budding off. So if you think about ER in the video, it was this blob, and then once in a while you would see this bulge in it, and then a vesicle buds off. That somewhat helps curve the outer surface and have a few more outside. But if the vesicle is not ready to bud off, there's an enzyme called flippase, which is my favorite enzyme name ever. Flippase takes some from the outer leaflet and moves them to the inside. I want to go back to this movement slide. I didn't talk about the flip-flop. Membrane lipids move laterally, they flex, they rotate a lot. They don't often flip-flop. Why would that be? Why do you need an enzyme to flip-flop? It takes energy. Yes. Why would it take energy to make? Why would they not do that on their own? To do that requires a hydrophilic head to pass through the massive hydrophobicity of the interior. That's highly energetically unlikely without an enzyme to force it. So once a membrane is made, they hardly ever flip-flop, but once a bilayer is made and in place, there is not usually flip-flop unless you have flippase to move lipids from one side to the other. So where is flippase? Where do you find it in the cell? In the ER. Well, associated with the ER. I think it actually stretches through a membrane, move some from one side to the other. You would have to be in the membrane to do this. Not shoving from out here. Get over there.
I think I'm confused. So when it's being made in the ER, are like the new phospholipids getting transferred to like the like plasma membrane of the cell, or like?
Not yet,
Okay.
not yet.
Okay.
We haven't talked about that yet, and I'm glad I still have this over here. So that's a good question. Any membrane lipid that is in a cell, it was originally formed in the ER, like this. And say if it's supposed to, if it's the type of membrane lipid that's supposed to go to the plasma membrane, it will get there in a vesicle. And we haven't talked about vesicle trafficking yet, but we do, I think, in the next unit.
Okay.
So what would happen is several plasma membrane lipids, those are kind of the default. Like those are the most important ones for the ER to make. They would get put into a vesicle and get sent out to the plasma membrane. Or if they were going to a lysosome, that vesicle would have lysosome variety. And it wouldn't be the same vesicle, it would be vesicles heading for the lysosome. That's a little bit related to this next slide too. So imagine, well, you don't have to imagine, they have it labeled. Here's the ER, that's where lipids are being made. They're sent to the outer leaflet, which is dark orange. FlipAce can send phospholipids back to the other leaflet if we need balancing. But if these lipids or lipid bilayer is supposed to be headed to a new destination, buds off into a vesicle. The vesicle goes to the new destination. It knows where to go. We'll talk about that later. And it will fuse with that membrane. So, pinching off a vesicle takes energy and a certain set of proteins we talk about later. Once the vesicle forms, I want to add this. Once the vesicle forms, it moves along microtubule roads. I'm definitely going to show that video after the break. It moves along microtubules carried by motor proteins, and it heading to its destination. It gets to the destination membrane and it fuses. So we've got membrane fusion here. But this vesicle needs to be carrying, when it fuses, it's got to fuse with the direct orientation. And this is really important concept. So, all right, here's the cytoplasmic membrane. Tan is the cytosolic phase. Maybe if you've got black and white slides, you call it light gray. Call it light. The light color is the cytosolic base. And the dark color, this is wrong. It's the extracellular phase. The dark color is the cytoplasmic phase. Make sure you write that down correctly or visually understand it. So out here, the cytosolic phase, the extracellular phase is light colored. The cytosolic phase is dark colored. If a vesicle arrives there and fuses, it's got to maintain its correct phase orientation. So notice here, the dark is the cytosolic phase, so it is over here. You've got the cytosolic phase and other. So over on here, almost every compartment in the cell has a membrane that has one face facing the cytosol. So lysosomes would, ER would, Golgi would. Cytosolic phase must always be the cytosolic phase because of the way membranes fuse. So a vesicle, the outer leaflet of a vesicle must be the cytosolic face, and the interior of the vesicle is the other. If this is going to the plasma membrane, the other becomes the extracellular face. And what this means is that whatever was carried on this side is released on this side too. Does that make sense? Yes.
So does the plasma membrane that's facing the extracellular space, does it just get longer? Because it fuses?
It does not get infinitely longer. This would make it longer. So vesicles fusing, which is also exocytosing what was in them, right? It would make the plasma membrane out here longer. But the cell is also constantly bringing in some membrane
Okay.
as a way to get rid of old phospholipids and recycle. So let's see. I could put that on here. So there's vesicle traffic out and there's vesicle traffic in.
So it kind of keeps things.
Yeah, it keeps the cell from getting too large.
That plays into the import-export.
Yeah. Like I have to talk about membranes here, and then import-export comes later. Probably be good. There's a slide nicer than this of this process that would probably be good to put in. And that answers a lot of questions. The other thing I want to draw onto this picture that's really important is that if we have phospholipids here that have sugar chains, so I'm drawing a sugar chain. Sugar chains that are made in the interior of the ER end up on which side of this? Out. They don't change shape. I'm not good at drawing. So they stick out. At no point do these ever see the cytosol. They're either interior to the ER, they're interior to the vesicle. Draw it here too. They stick out here. I think this is a good time for a break. So if you'll hit that.